Hello and welcome. My name is Floriana Blanton and I am an outreach coordinator with the Cornell Laboratory for Accelerator Sciences and Education. The activity that we are going to complete today is the isolation of DNA from your own cheek cells. Let's go ahead and review the materials that we are going to need before we get started. If we were at school or at a lab, you would be using measuring instruments such as graduated cylinders, beakers, or pipettes to get the amounts that you need. But because we are all at home, we will be using common household items to obtain the amounts that we need. You will need one transparent cup, which will be your experimental cup, half a cup of water, which is about 100 milliliters of water. You will need 2 tablespoons of salt, or about 28 grams. You will need 2 drops of any dishwashing detergent, some toothpick, about 1/4 of a cup or 60 milliliters of rubbing alcohol, and a timer. I will be using my phone as a timer. Before we get to the procedure, there is one last thing we need to do. We need to talk about the DNA, its importance, its chemical structure, and its position inside cells. This is a model of a DNA molecule. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. They pair up in a very specific arrangement in which adenine pairs up only with thymine and cytosine pairs up only with guanine. Each base is also attached to a sugar molecule and a phosphate molecule. Together, the base, the sugar, and a phosphate are called a nucleotide. And the nucleotides orient themselves in a very distinctive double helix pattern. Human DNA contains about 3 billion bases and has a length of about 2 meters, roughly 6 feet. And where is DNA located? In the cells of eukaryotic organisms is located inside the nucleus. Here we have a very colorful model of an animal cell. The DNA is located inside the nucleus of the cell. That means that for us to be able to access it, we'll have to break both the cellular membrane and the nuclear envelope. There will be steps in our procedure that will take care of these 2 structures. Well, before you get started, the first thing that you're going to do is measure about 1/4 of a cup of rubbing alcohol, the alcohol that you can get in any pharmacy that you probably already have at home. And you are going to place it inside a container that has a lid. That container needs to be properly labeled alcohol, and then you are going to place the alcohol inside the container in your freezer. Please do not put anything in your freezer that is not labeled accordingly. The first step is to place all the salt inside the container with a 1/2 cup of water and mix it with one of your toothpicks until it is completely dissolved. It is now time to collect the cells that we are going to extract the DNA from, and those are your cheek cells. Take about 15 seconds and gently, very gently and carefully, scrape the side of your mouth with your teeth. It's gonna look like this. Do that for 15 seconds on one side and then switch to the other side. Do not swallow the saliva because that's where your cells are. After 15 seconds on each side, you are going to take the salt solution and place a small amount inside your mouth. You're going to swish back and forth, and then you're going to put that product inside your experimental cup. We now have a solution that contains both your cells and the salt water that we prepared before. But we haven't really done anything to get to where the DNA is actually located. If you remember from the introduction, we pointed out to the position of the DNA inside the nucleus of the cell. It means that in order to get to it, we're going to have to break the double lipid, the double fatty membrane of the cell, as well as the fatty membrane of the nuclear envelope. And we are going to do that by adding about 2 drops of any soap, absolutely any dishwashing detergent that you have in your home, to the solution that contains your cells. At this point in time, we will be breaking all the membranes in order to try to get to the DNA. So from here on out, anytime that you mix this solution, you are going to be very careful because you do not want to break the long strands of DNA. We are going to let the solution sit undisturbed for a couple of minutes while we go over what we have done. In the first step, we prepared the salt water. In the second step, we collected the cells from our cheeks. We use cells from the cheeks because your body is constantly replacing them, and so they are a great source of DNA. We added a little bit of soap in order to break the membranes and get to where the DNA is located inside the cell. In the next step, we are going to be precipitating the cells that are already in our solution. It turns out that DNA is soluble in water, but is not soluble in alcohol. So you will retrieve the alcohol from your freezer, and we are going to, again, very carefully add it to the solution with the cells. The DNA strands are already in your solution, even if you cannot see them. So in the next step, very carefully, we're going to add the very cold alcohol into the experimental cup with the DNA strands and the salt solution by pouring it slowly and gently on the wall of your cup. Very carefully, slowly, gently. And then you can look inside the cup where you will see the strands that are already precipitating. That's your DNA. We needed to cool down the alcohol in order to increase the chances of the DNA precipitating. As you now know, DNA is not soluble in alcohol, but if the alcohol is cold, it increases the probability that the DNA will precipitate. We use salt for a very important reason. In nature, DNA is negatively charged. If we release the strands of DNA into plain water, they will be repelling each other because of the charge. When we dissolve salt, sodium chloride, in water, the positive sodium ions will neutralize the DNA's negative charge and allow the strands to form clumps.